Roots can only take up water and some small minerals, but roots cannot take in the one element that all life needs to survive: carbon. Carbon is a brick in every important cellular structure and is responsible for most of the organic matter in the world. Without carbon, plants and indeed life cannot really exist. The easiest way for plants to get their carbon isn't from the soil, but rather from the carbon dioxide in the air. Plants have an elaborate process to convert that carbon dioxide into a usable food source that we all know: sugar. This critical process is called photosynthesis. You can think of photosynthesis as a factory production line. This factory is located in separate organelles called chloroplasts, which are mainly present in the leaves of the plant. If one looks at a leaf through a microscope, one would see many tiny green blobs within the plant cell. These are the chloroplasts. Like any other factory, the chloroplasts are well organized. For photosynthesis, there are two important areas in the chloroplast. One area generates energy in the thylakoid, while the other acts as the assembly line in the stroma. The first step is to gather the energy given by light, primarily sunlight. Plants have built-in solar panels to achieve this. For almost 3 billion years, life has been using the sun to fuel its machines. However, plants can't directly use the energy from the sun to power their machines. They convert this energy to something they can use: ATP, the energy currency of the cell. The area in the chloroplast that generates the energy for the machines is the thylakoid. Thylakoids are those green discs stacked one on top of the other in a tower shape. These thylakoids are like the plant's personal solar panels. In the thylakoid's membranes, there are many pigments that absorb the energy of light at different wavelengths. The most important of these is chlorophyll A, which absorbs light in the red and blue region of the visible spectrum. Once these pigments absorb the light energy, they pass this energy to certain large protein structures called photosystems in the thylakoid. These photosystems begin using the light energy to convert it into a more usable form of energy for the plants. To begin converting this light energy into usable energy, photosystems have a special chlorophyll A molecule called the reaction center, which absorbs light of a very specific wavelength. This special chlorophyll A pigment is denoted by the wavelength at which it absorbs light rays. Scientists use different nomenclature to distinguish chlorophyll A from other pigments. The chlorophyll A of the reaction center is suffixed with a P and the number of the wavelength of light it absorbs. Plants have two photosystems: PS1, which has chlorophyll A P700 and absorbs light at 700 nanometers, and photosystem II, which has chlorophyll A P680 and absorbs light at 680 nanometers. These two photosystems, along with a few other proteins, work together to harness the sun's energy. When light hits photosystem II, water is split into oxygen and 2 protons, and gives an energized electron to the photosystem. Photosystem II takes some energy from the electron and passes it to another protein complex called cytochrome Bf complex. Cytochrome Bf carries the electron to the other photosystem, photosystem I. Light energy absorbed by chlorophyll A P700 in the photosystem I energizes the electron again, which allows PS1 to use that energy. In this process, it is essential that the photosystems replenish their electrons. This is achieved by water. Without replenishing the electron of the reaction center, the entire process will come to a halt. The work that photosystem II and cytochrome Bf do with the energy is transporting protons from outside the thylakoid into the thylakoid lumen, the space between the two thylakoid membranes. This creates a difference in the chemical concentration of protons between the membranes, and since the proton is charged, an electrical difference is also created. The plant uses this electrochemical gradient to create ATP molecules. There is a special protein complex present in the membrane of thylakoids called ATP synthase, which opens a door for protons to exit the thylakoid lumen. As protons leave through ATP synthase, they power the production of ATP. In this way, light powers the plant factory that makes the food. Since all these processes require light, they are called the light-dependent reactions, or simply the light reactions. Now that energy is available, the machines can begin building the food. These machines are called enzymes and are critical elements in all life forms, as they help to carry out different chemical reactions in a cell. Plants use a host of different enzymes to help manufacture their food. This brings us to the stroma of the chloroplasts, where the food-making enzymes are found. Carbon dioxide is taken into the plant cells through openings in the leaves called stomata. Once in the stroma of the chloroplast, the carbon dioxide enters the assembly line to be converted into food. This starts with the enzyme called ribulose 1,5-bisphosphate carboxylase, or Rubisco for short. This enzyme takes a starting material called ribulose 1,5-bisphosphate, a 5-carbon sugar, and joins the carbon dioxide to it, and then chops the resultant 6-carbon molecule into 2 3-carbon molecules called 3-phosphoglycerate. This molecule is further converted into glyceraldehyde 3-phosphate in 2 steps involving 2 different enzymes. Those 2 steps require 2 ATP molecules and 2 NADPH molecules. The 2 glyceraldehyde 3-phosphates can now join together to make sugars, such as glucose, fructose, etc. These sugars can be used to make other molecules such as starch, which is plant food, cellulose, which makes up the plant cell wall, sugars to make DNA and RNA, as well as fats and proteins. We use these same sugars to fuel chemical reactions in our own body. Even the proteins and fats present in plants can be made by chemical reactions using these foundational sugars. Other photosynthetic organisms, such as bacteria, algae, and protists, perform similar reactions to survive. Without this ingenious piece of natural engineering, life might not have expanded in size and complexity as it did. This cycle also balances the amount of CO2 in the atmosphere. As life developed the ability to photosynthesize, these organisms removed CO2 and pumped generous amounts of O2 into the atmosphere. This made it possible for life as we know it to evolve. As forests continue to be burnt, all the carbon dioxide they store in their plant matter is released back into the air. With diminishing green cover, there might not be enough plants to remove all the CO2 that we're carelessly pumping into the atmosphere. The fact that life evolved such complex but elegant processes is indeed marvelous. At its core, learning the biochemistry of life is all about observing the elegant solutions that nature has developed, solutions that no feat of human engineering has yet to rival.